So this is the second part of Unit 3, and this is plant and animal breeding. So we're talking about food in the previous topic, specifically related, plant life, I suppose, and because we're looking at photosynthesis and the need to try and get enough food to enough people as we have a increasing human population. So this topic, this next part of Unit 3, we're going to look. So first part of this, describe how plants and animals can be improved by breeding. So first of all, just a definition. So we have selective breeding. So this is a process which is controlled by humans. So it's breeding plants and animals to produce new or improved organisms as sustainable food sources. So sustainable, again, a key word here because it's something that we have to maintain for a period of time. So, for example, selectively bred. I mean, a lot of our food is. So, if you look at these, I don't know how many of these you like. I think I like all of them. Cabbage, cauliflower, and so on. These have all been selectively bred from one original plant species. And then over here we have cattle, which we're going to just a bit talk about in more detail. So, are you ready for this? Let's play Name That Cattle. So put down numbers, I think it's maybe 1 to 7, and I want you to tell me what each of them is. Let's see if you can get 7 out of 7. Any farmers? What is that? That's number 1. So, I mean, well done if you got this. This is called an auroch, and this is extinct. So this is a wild cattle species which became extinct in Europe in the 17th century. So if you went back a few thousand years, this is what you would see. You wouldn't have the kind of cows that we see just now. You would have aurochs, and they are the ancestor of all domestic cattle. So they were domesticated a few centuries ago. Might get easier from now on. Here's number 2. So this is a bull. I think you might have got that. What you might know is just the kind of differences between these different types of cattle. So a bull is defined as a non-castrated male. Okay, so a male which is still capable of being able to produce offspring. And yeah, there's not too many of these around, obviously, because they can be quite aggressive. Number 3. Oh, this one's called Daisy. So number 3, this is of course a cow. So a cow, scientifically, is a mature female, and yeah, they've been selectively bred. So you can see here a lot of milk can be produced, and within a day, they can produce 22 litres of milk. However, essentially they need to be almost continuously pregnant, I suppose. So they need to have a calf every year in order to keep up the milk supply. That's why you would have, one bull on a farm and a large number of cows. 4. These are calves, and that is just that's unweaned cattle. So these are cattle that are still taking milk from their mother. 5. I think this is a term you might also apply to horses, if that helps. So these are called yearlings or stirks, and they are weaned calves then, so they tend to be about 1 to 2 years old. Number 6. What we have here are a heifer. So a heifer is defined as a female who hasn't given birth yet. Okay, and then the last one, 7. That's quite a big animal, isn't it? Any ideas? That's a bullock. So a bullock is a castrated male, so it's not going to produce any offspring. And the reason why it's the reason why humans have it is to raise it for beef. So if you have your minced beef, this is where it's coming from. So bullocks. Was that good? Did you enjoy that? Oh, hang on, there's another one. Right, last one. This is the last one. So this is number 8. So these, again, you will know this. This is an ox. So not because we've kind of moved to mechanical forms of ploughing and so on. They're not really common in this country, but certainly in the developing world, we see a lot of oxes. So an ox is a castrated male, and it's used for, the technical term is draft work, so that is pulling loads. So that's the kind of range of different cattle that we have. So I mean, I guess if you're out and about, the most likely thing you'll see is a cow. But yeah, all descended from oryx, and all have been selectively bred for different jobs, for different functions, whether it's for food, whether it's for work. So that's cattle that humans have selectively bred. The other obvious one, all dogs are descended from wolves, and they are the same species. They're all dogs, but what we have is different breeds. And as a result of selective breeding, humans have made them look different. And this is something which has only happened in, the domestication of wolves probably happened at the time that humans started farming. Which we're talking about the last 10,000 years or so. So dogs are pretty, recent addition to life on the planet, I suppose. And you can have a little think of this. So which dog breeds would you mate for the following roles? Guard dog, waterfowl, retriever, search and rescue. So the guard dog, there's different ways you can do this, but you might think, yeah, very, very loud bark, maybe combined with sight, perhaps. So maybe you'd want to go for this one, breed F with breed B, perhaps. Waterfowl retriever, so they would have to be maybe speed. You'd want something that was above average. You'd maybe want, I don't know, a shorter hair length if they're going into the water potentially. Good sight and smell as well. And search and rescue, maybe kind of similar for that as well. Maybe hearing for them would be particularly important. Search and rescue missions might go on for a long time, so maybe endurance. So these are the things that humans will have thought about when they've bred dogs, because they'll have been bred for specific purposes. And yeah, have a look at this. So evolution of dogs explained. So it just goes back to that sort of speculating about what the circumstances were which led to people in the past, I suppose, would have been wary of wolves and potentially even scared of them, and certainly they've been hunted to extinction in the UK. But there's been a period of time where people have taken them in and taken them into their homes, basically. So this kind of talks through what might have occurred. So selective breeding, we haven't really talked, we've kind of talked using examples about characteristics, but see if you can do this. So describe at least 2 characteristics which breeders might seek to improve in plants or animals. And we're being specific here, so we're talking about sustainable food sources. So see if you can write down a couple of things you might want in terms of plants and animals if they were going to be used for food. So press pause. Okay, so the characteristics you don't want to improve. So crops or animals with higher food yield is the word. Higher nutritional values as well. So you can get enough, even if it's maybe a small amount of food that's rich in the vitamins and whatever else that humans need. Something else that would be useful would be resistance to pests and disease. And finally, and this is a kind of big one for the future, is ability to thrive in particular environmental conditions. So if you have a period where there's no rainfall and all your crop dies, then maybe you might want to think about, should we go down the route of having genetically modified crops which are able to survive in lower in conditions in which there's less water. And you can have a look at this as well. So this is the history of food. So, we talked about going back to the domestication of wolves, and if you go back maybe 10,000 years at the end of the last ice age, most people were hunter-gatherers, so they just moved around, they followed animals, they knew where berries were, and they would eat them at certain times of the year. But then there was a period in time when humans decided to settle and they domesticated plants, and from that you had villages and then towns and cities and so on. So have a look at this history of food. And that is just the introduction to this, so have a break and then we'll come back and we'll look at kind of trials you would do if you wanted to selectively breed plants. So this is the second part of this. And a bit of a starter to see if you remember what you did last time. So describe 3 characteristics which breeders may seek to improve in plants or animals which are to be used as a food source. So write that down. So what you should have is any one of these. So you want a higher food yield, so that might just be simply larger crops. Higher nutritional values, resistance to pests and disease, and finally the ability to thrive in particular environmental conditions. Or you could have put it a bit more simple, you could have been specific there, you could have said be able to grow in areas where there is low rainfall. Okay, so the next part of this, so if you're trying to produce these plants which have higher yields or have higher nutritional values, what you would do is, these days you would set out plant field trials. I mean, going back to the early humans, they wouldn't have, selective breeding wouldn't have been, they wouldn't really know the science of it, but what they would, if there was a particular plant that survived a frost, then maybe they would, they would take the seeds from that, or if there was a particular plant which grew more than the others, then they would take the seeds. So kind of basic intuition, but now you can have plant field trials and try and produce crops with characteristics that you want. And then after that, we're going to have a look at inbreeding and then inbreeding depression. So plant field trials, so what you do is you carry out, it's a bit like, think about, it's almost like you're having different experiments and different test tubes, but you're using different fields, I suppose, or different areas of fields. So plant field trials are carried out in a range of environments to compare the performance of different cultivars, so that's just cultivated crops, or treatments, and also to evaluate genetically modified crops. So what you have here is this area, this is basically, this is kind of like an outdoor laboratory, isn't it? So you've got different sections which are separated, and you will, change, as you would in the laboratory, you'd maybe change one variable in each one and see what's produced in terms of plant growth and resistance to disease and so on. So let's just kind of take an example. So if you're designing a field trial, so let's imagine you're investigating the effect of different fertilizers. So we're saying fertilizers A, B, C, D, E on crop growth. And here is your field. So you've got plot 1, plot 2, and plot 3. So there's something going on here which is not particularly scientific. So have a think about what that might be. Any ideas? So what's the aim? To find out the effect of different fertilizers A to E on crop growth. So the problem here is that they've investigated 2 things, haven't they? So they were just looking at the effect of different fertilizers, but they've also changed the concentration of the fertilizer. So there's more than one variable altered. Okay, so that's the first thing to talk about when you're designing a field trial. So beforehand you want to make sure the selection of treatments is, you need to consider that in order to ensure fair comparisons. This is your experimental design basically. The second one again, so we're looking at fertilizers A to E. What are they going to increase? Which one increases crop growth the most? What about this one? Any issues with that? So with this one, each fertilizer is in the same part of the field. So it could be that, this could be quite a distance between these two areas, and it could be this area gets more sunlight. It could be that this area here, could be a bit of a slope. So down here you get more water. So that would, again, if someone was to question your experiment, that would be an issue. So what you'd want to do is kind of randomize this. So that's the second part. So something you need to consider beforehand is also randomizing treatments, and that's in order to minimize bias when you're measuring treatment effects. And that's the last one. So issue with that. So this is something that probably, going back for a few years now, you would have come up with this because we talk about it from S1, I think. So the experiment here has not been replicated. So the last part of this, if you're designing field trials, is to have a number of replicates, and that is to take into account variation and, as we always say, to improve the reliability of results. So ideally, this is what you'd want. So you've got concentrations not mentioned, so we assume that it's the same as being replicated. So you've got your 3 for each different type of fertilizer, and it's randomized. Okay, so there's your ideal design. Appropriate treatment selected, randomized, and replicated. So the next part of this, before we talk a wee bit about genetics, I just want to make sure we're okay from National 5 because some of these terms will come back up. So from National 5 revision, we'll say that an individual has got 2 copies of the gene. So the gene for here is pea plant shape. So that each copy is known as, what's that? Allele. So 2 copies of the gene, alleles for each characteristic, and you get 1 from each parent. So do this, press pause, and just match these up just to make sure you're clear on what these terms mean. So number 1, a gene is A. So that's a section of chromosome with a DNA sequence which codes for a protein, and then the protein controls a characteristic. Allele we just mentioned, so that is the different versions of a particular gene, so that's D. The phenotype is G, so that's the physical characteristics of an organism. Genotype, number 4, is the letters. You usually write it as letters, so the letters for an individual's alleles for a particular characteristic. And then dominant, so that is an allele which shows if there's only 1 copy of it. So you get 1 allele from your mum, 1 allele from your dad. If you had, say, the brown allele for brown eyes allele from your dad, and you had the blue eyes allele from your mum, you would have the phenotype brown eyes. So the brown eyes is dominant. And 6, recessive. So that's C, an allele which only shows when there are 2 copies of it. So if your sister had blue eyes, then that would mean that she's got 2 copies of that recessive allele from, 1 from your dad and 1 from your mum. And then 7, heterozygous, that's when 2 alleles of a particular gene are different and often written as capital letters, so it could be big B, small b. And then homozygous, that's H, A is H, so that's when the 2 alleles of a particular gene are the same, so that would be big H, or small h, and that's also called a true breeding genotype. Okay, based on that, try this as well. So this is 9 to 13. So this is the individual. Each of these, what's that? Each of these, what's that? And yeah, try and fill it in. So number 9, these are, because it's the letters, that's the genotype. Number 10, the actual characteristic, what you see, that's the phenotype. If that's big B, then 11 is homozygous. This one here, 12, also homozygous because they're both the same, both alleles. And then this one, the alleles are different, so 13 is heterozygous. And then just 2 more to have a go at. So big B, brown eyes, is the something allele, 14 is dominant, and then small b, blue eyes, is the 15 is recessive allele. Okay, just some of these terms that come up again, so that was just a bit of revision. So inbreeding, yeah, so description, example, advantage and disadvantage. So we can write down this table, press pause. So in the table, I want you to use this information to fill it in. Okay, so press pause and do that. So what you should have, just a definition of inbreeding, something along these lines. So inbreeding is when related individuals are bred together for several generations. And an example would be dairy cows with high milk yield. And then the advantage is that you end up with homozygous individuals and they have the desired characteristics. But the disadvantage is you also get individuals who are homozygous for recessive deleterious, so those are harmful in some way, deleterious alleles and are unlikely to survive to reproduce. So this is inbreeding depression when you have these unwanted characteristics as well. Okay, so inbreeding positive because in terms of breeding plants and animals, because you get these desired characteristics. However, it does have its downside as well. You can end up with inbreeding depression. Okay, let's have a look at some examples of inbreeding. So not necessarily for food species, but you might be, royal families in the past, inbreeding has occurred. People marry their cousins and so on, and this is one of the kind of famous examples of that in Europe. So this is the Habsburg jaw, and this family, so they ruled Spain in the 1500s and 1600s, and yeah, as I said, common for family members to marry their cousins, and yeah, they reckoned that these are kind of homozygous recessive issues here. So some family members had jutting jaws, bulbous lower lips, long noses, and large tongues, which would cause problems with eating. So if they had that natural kind of outbreeding, this probably wouldn't have occurred. So thought to be the result of the passing on of a recessive gene. The second one is haemophilia. So this is in descendants of Queen Victoria. So again, it's royal families again. And what you have here, if you go back to Queen Victoria's ancestors, the ones highlighted in red are haemophiliacs. And if you look at the dates, how long they lived for, he died when he was 31. Prince Friedrich, unfortunately, was just 3. What's that? 33. He made it into his 50s, 4 years old. So a lot of premature deaths here. And so what happens if you're a haemophiliac is that your blood doesn't clot as it should. So it's resulted in prolonged bleeding. But again, in normal circumstances, this would not be, it wouldn't run through families because there would be, new genetics would be brought in from unrelated families. So afflicted many of the male descendants of Queen Victoria, and then her son as well. He died of a brain hemorrhage after a fall, and other descendants died prematurely as well after events such as falls, operations, and car accidents, just because basically they bled and they couldn't stop the bleeding. A third one, these children here, you might be familiar with this. This is in America. So these are the Amish. So religious communities, they live a simple farming life in America, and they tend to marry within the community. And there does seem to be a positive side of this, although it could be debated as to whether this is maybe due to diet. So they have lower cancer rates than the general population. However, they do have various genetic diseases. So one of them is Angelman disease. So children don't have a kind of delayed development. So they might have speech impairment, often misdiagnosed as autism. And something else is that unusually. There's a higher incidence of dwarfism than the general population. So again, two kind of in the general population unusual conditions, but they tend to occur more in the Amish, and it's thought to be because of inbreeding. So watch this film, What Happens When You Inbreed. Search for that on YouTube. And then time for a break, and then there's one more session in this selective breeding part. So to finish off, just before we get on to the last part of this, let's do some revision of the last session. So put down numbers 1 to 3 and see if you can match those up. Press pause. So this is when we did our field trials, and we had the example was if you were using different, you're trying to investigate the effects of different fertilizers, and you would want to have an appropriate selection of treatments in order to ensure fair comparison. So one was B. 2, randomized treatments to minimize bias. And then 3, replicating the experiment, and that's to take account of variation and also to improve the reliability of results. Second bit of revision, press pause, put down numbers 4 to 10 and see if you can do this summary of inbreeding. So inbreeding, the description is number 4, so it's related individuals are bred together for several. Number 5 is generations. So an example would be dairy cows with high milk yield. And then the advantage 7 is homozygous individuals are produced with desired characteristics. So produces individuals who are homozygous for number 9, recessive deleterious alleles and unlikely to survive to reproduce, and that is called inbreeding depression. So last part of this is to explain why plants and animals are crossbred and what F1 hybrids are, and then finally everything we've talked about is, well, I suppose it's natural in the sense that there's no kind of technology involved. Humans have just sort of manipulated who's going to be which animals or which plants to breed, to produce offspring from. The last bit is to describe how genetic technology can be used when in breeding programs. So again, just a wee bit in that 5 revision, so we know what we're looking at here, and when we talk about F1 generations. So we did, you remember this hopefully, so you do monohybrid crosses. So if you've got parents here, there are their 2 alleles, so they're both homozygous for their particular characteristic. This one homozygous dominant, this one homozygous recessive. So the gametes that are going to be, in this instance, bred together are big H and small h. So this means that the whole of the F1 generation, big H times small h, gives all big H, small h. So this means that all the offspring are going to be curly hair. Then to work out the F2 generation, you do this little Punnett square. And when you have a go at that if you want, when you do the Punnett square, you get this. So what you can see is that when you get to the F2 generation, you start to have that variation again. So the grandmother, if you imagine this is the grandmother, has got straight hair, and then the parents don't have straight hair, but the straight hair can reappear again because the allele is being passed on. Okay, so you've got a parental generation, then you've got an F1 and you've got an F2, and this F1 generation are the ones we're going to look at next. So you have crossbreeding animals. We talked a wee bit about it related to dogs, didn't we, that little exercise? So it's just when different breeds, and they're often inbred, produce a new crossbreed population with improved characteristics. So for example, you might want to breed together a high meat yield with a high lambing rate. And what you would want to do is the 2 parent breeds, you keep them going, so you can also breed them amongst themselves so that you can produce more crossbred animals. and that, if you could think back to our monohybrid cross, the F1 generation, they gave you this heterozygous generation. They were all heterozygous, so you cross these two inbred lines and you get this heterozygous crop, and it will have increased vigor and yield, so it is desirable. Okay, and you'll see these actually advertised. So F1 hybrid plants, great for showing and eating, easy to grow, etc. There is a problem though. So if you've got the F1 hybrids and they have characteristics which are desirable, then if you then breed the F1 hybrids, then you get that F2 generation that becomes variable again. So you begin to see characteristics which you do not desire. Okay, so this is why the F1 hybrids are not usually bred together. I just put this in as well because this can occasionally comes up as an exam question and can cause a wee bit of confusion. So just to talk you through it. So triticale is I don't know if that's how you say that, but anyway, it's a hybrid cereal species which was produced by crossing Triticum durum, a species of wheat, with Sicale cereale, a species of rye, as shown in the diagram. So you've got N here, and again, I'm doing this because you might not remember that N is the number of chromosomes. So you've got two sets of chromosomes here, so it's diploid, and 2N is equal to 28, so that means that N is equal to 14. This is bred with this, and they're not going to tell us this because that's the question. Then you've got an F1 hybrid, which is sterile. However, you can artificially double the chromosome number, and if you do that, then triticale 2N is equal to 42. And the question is, what was the diploid 2N number of chromosomes in Sicale cereale? I'm going to let you just have a wee think about that, so press pause and I'll tell you the answer. So if the triticale at the end, 2N is equal to 42, and the chromosomes number was artificially induced to double, then that means, I'm going to try and draw here. Oh, that works. So 2N here, well, that's a bit big. Oh, I'm not very good at this. So 2N should be 21, okay? If 2N is 42 and the chromosome number has artificially doubled. Now, they're going to get 1N from this one, so they must have got 14 from this, which means that if that's 21, they must have got 7 from Sicale cereale. If they get 7 from that, then, and that's 1N, then 2N must be 14. So the last part of this is, looking at technology. So with sequencing now, you can use that to identify organisms with desirable genes. Okay, so you might not necessarily need to wait and breed them. So you might be able to recognise through genetic sequencing that you've got an organism with, for example, disease resistance. So one of the desirable things we talked about, nutritional value and so on. And you can use genetically modified crops as well. So that's, remember, going back to Unit 2, you've got your recombinant DNA technology. So if you think about a pest, so maize, which is sweet corn, so you can add Bt toxin gene to their genome and they kill caterpillars. So the caterpillars, of course, would reduce the yield of the crop. So that would be a possibility. GM crops, by the way, they're quite commonly used in America, but not in Europe at the moment. Or I don't think in most of Europe. Not for food for human consumption anyway. Maybe for feed for cattle. So just to give you one possible example, so turbocharged rice. So this would be again, the growth of plants. Remember, from photosynthesis, it's dependent upon that enzyme Rubisco. So what you can do in these plants is they can be genetically modified so that carbon dioxide concentrates around Rubisco. I don't know exactly how that occurs, but this is what happens in these genetically modified crops. As a result of that, the rate of photosynthesis increases, so you get more crops and a higher yield. and you can feed more people, I suppose. So in this instance, this is an example called turbocharged rice. And that is the end of this topic. So we've been through reasons why you would want to selectively breed crops, and then we've looked at problems with with inbreeding, also how you would design field trials if you're trying to produce these desirable crops, and then we finished off by talking about kind of F1 hybrids, why they're desirable and why you would want to keep that generation, and then finally what you can use, what you can do in terms of genetic technology to improve and what you can do to provide food for people.